So as Leah said, I'm the field application manager at IDT, covering CRISPR products as well as our functional genomics products at IDT. And those include things like siRNA and antisense oligos. My job really is about supporting our customers, whether that's experimental design, troubleshooting and technical support, or just helping them purchase products for more custom projects that they can't really order through the website. So I've been with IDT nearly 5 years now, and today I want to talk a little bit about our R&D efforts to understand really robust ways of quantifying editing, both at the intended editing site, the on-target site, as well as off-target sites using CRISPR. So I'll turn my camera off for the remainder of this, just so you don't see me looking in multiple directions. But as Lee and Marshall said, if you have any questions, please do enter them in the Q&A, and we'll get to them as we get to that part of the talk. So as you can see on the slide here, we're going to be discussing the use of our rhAMPSeq products for CRISPR. I want to start a little bit higher level though, and just sort of refresh everyone's memories about IDT and really what it is that we do. So if you're not familiar with us as a company, it's good for you to know we've been around a little over 30 years, and our origins really stem back to custom oligo manufacturing. So the picture that you're seeing here on the right is one of our labs in our main production site in Iowa in the US. And each one of these machines here you see is actually synthesizing 96 oligos at a time. And so you can see we have quite a bit of capacity for this. Currently, today in the sort of post-COVID world, we'll call it, we're making about 64,000 oligos every day, although our capacity is substantially higher than that. Many of those will go out to customers like you for use in all of your experiments, but a good number of them stay internally at IDT and either become other products, right, like our G blocks and synthetic genes, or they go to our research teams. And I mention them here because really I'm introducing work that they have been working on for many years. So we have 15 or 20 dedicated R&D scientists solely working on CRISPR related projects at IDT. So all the data that I'm going to show you today really comes from their efforts. I'm not going to talk too much about CRISPR technology today, or really the CRISPR products we offer, but I did want to just quickly flash this up here so you're all aware of the spectrum of what we do in the CRISPR space. We like to think of ourselves as really supporting most of the CRISPR workflow. So that obviously starts with guide design, and if you're doing a gene knock-in experiment, donor template design. We have lots of protocols that we've developed or that our collaborators have developed for us for optimized delivery of those reagents. We have a whole range of guide RNA formats that will be useful for different types of experiments, as well as a range of proteins to support those. We'll talk a little bit actually about this high fidelity Cas9 later on in the presentation. A lot of the that I'll share today were used to characterize how this protein reduces off-target editing. So that's relevant for today. Lots of options for donor templates as well, depending on the size of the insert you're trying to knock in, if you're doing a knock-in experiment. And then the bulk of today's talk will actually focus on the analysis section that comes after you've done a CRISPR experiment. So we have some PCR-based methods like a T71-based assays, really quick and easy ways of detecting if genomic editing has occurred. But we're going to focus on rhAMPSeq, which is really probably, of all the options we support, the most robust when it comes to quantification and the ability to generate sequence data. My intention today is not, like I said, to talk too much about how CRISPR itself works. I assume most of the people in the audience here understand the basics of CRISPR and what all the various components are doing once you get them into the cell. But I do just want to quickly talk through this slide to set the stage and make sure we all have a shared understanding of the terminology. And so really a CRISPR experiment starts with what you're seeing on the top half of this slide here. We're showing an example of either a Cas9 ribonucleoprotein complex, so RNP, or other CRISPR nucleases like CPF1 or Cas12. Of course, these would also be combined with guide RNAs. And once you get this into the cell, it's going to seek out a specific location in the genomic DNA based on the sequence of the guide RNA and introduce a double-stranded break at that location. Once that break is formed, the following process, which you see on the bottom half of the slide, is really up to the cell. So the cell's natural DNA repair pathways. The most common one, and probably the easiest one to take advantage of, is what you see on the left here. This is non-homologous end joining. And so this cellular pathway basically takes the blunt ends of the cut genomic DNA and ligates them back together. And occasionally that process will result in small insertions or deletions at the cut site. And when you do this in a protein coding region of a gene, you get a frame shift mutation and you knock out the gene. And so this is a really efficient way of knocking out genes, either one gene that you're studying or doing larger genomic screens. And in optimized settings where people have really figured out their transfection protocols and optimized the guide sequence, we can get nearly 100% of cells edited using this approach. One of the downsides, though, is of course you don't really control what the mutant genome looks like. The sequence of this indel is semi-random. It is reproducible, but it will be different from guide RNA to guide RNA. And you also can't insert much new genetic material this way. And so we see more and more interest in this pathway you see on the right-hand side of the slide, which is homology-directed repair. From an experimental standpoint, the key difference here is that you're including a donor template, which is some exogenous DNA that encodes for the new sequence you want to insert, shown in green. And we'll have homology arms, shown in light blue, which are homologous to the region adjacent to the cut site in the genomic DNA. And so when the cell repairs a double-stranded break using HDR, you actually get integration of that new sequence, and you have direct control over what the mutant genome looks like. So you could do things like introduce a premature stop codon, which allows you to not rely on the semi-random indels you see in NHEJ, or other things, right? Substituting amino acids out of a gene to study the effect of that, tagging genes with fluorescent proteins, or replacing entire genes or entire exons with different versions. So there's really a lot of potential here. It is a more challenging experiment just because HDR doesn't happen as often and it requires a donor template, but we see a lot of customers going this way. So that's a quick introduction to what CRISPR is doing. I think for the focus of this conversation, we're really focused on what's happening in the top part here, right? And this diagram shows the results of what we would call on-target editing, so double-stranded breaks that are happening at the intended site in the genome. But for the sake of today, we really want to sort of shine a spotlight on the fact that Cas9 and really every CRISPR enzyme is not perfectly specific. And so there's always a risk that it's going to bind to another site in the genome and introduce breaks there. And today we're going to discuss methods to identify those, and then more completely we'll discuss methods to quantify and sequence those. And so that's really what this slide is summing up here, is just that CRISPR is a fantastic tool for genome editing. It's very easy to program, which is really the reason it's so revolutionary, but it's not going to be perfect. Just like any molecular biology technique, you're going to have some non-specific events occurring. And so I think every person using CRISPR in their experiments needs to think about off-target editing. For some people, it's not going to be of much consequence. If you're doing large-scale screening experiments, well, yes, off-target editing is probably happening, but it's not going to be the focus of your efforts. But if you have ambitions to move your technology into, say, the clinic or as a therapeutic, you really need to have a robust way of characterizing any off-target editing. Because it's obviously a part of the safety profile of these kinds of things. So I want to start a little bit by explaining why we use this rhAMPSeq technology at IDT to do this. rhAMPSeq, and I'll go through more details about how it all works in the subsequent slides, but really rhAMPSeq is a form of Amplicon sequencing. So you use a highly multiplexed PCR to amplify all the on and off target sites in the genome that you want to study. You throw that on an Illumina sequencer, and then you have a robust way of getting sequence data and quantifying editing at all of those locations in the genome all at once. And so this is a really useful technology for experiments like this because it allows you to basically amplify all of those locations in one PCR reaction. So you get this done in about half a day and then throw it on the sequencer. So much faster and easier than, say, whole genome sequencing or other Amplicon sequencing technologies. And it also gives you much better sequence uniformity and on-target read mapping, which make the analysis downstream a little bit easier as well. One thing that I want to be clear on, though, is that rhAMPSeq is a technology for assessing editing at known on and off sites. it's not a technology for identifying where off-target editing is occurring. And that's sort of what we're looking at with that phrase at the bottom of the slide here. There are other techniques that I'll show you on the next slide that will help you identify where off-target editing is happening. And once you have that list of on and off-target sites, that's really where rhAMPSeq comes in. rhAMPSeq is a really highly multiplexed tool. So if you ever look on our website on how this technology works, you'll see we discuss designing libraries of up to 5,000 sites. So imagine a multiplexed PCR with 5,000 different amplicons in it. For CRISPR applications, you almost never need to have that many targets in your library. We typically see people using between, say, 10 and 100. But it's good to know that you can really expand it out quite a bit if you ever needed to. So to sort of explain that first step that I alluded to, when we talk about identifying and quantifying off-target edits, the first step is really identifying where they're happening. Now, if you've ever gone through a guide design tool or done guide design manually by hand, you're already doing this to some extent, right? So we've listed a few in silico prediction methods on the right here, including our own design tools and a few others that you'll find on the web. We've found that all of these tools do a very good job of flagging guide RNA designs that are risky. So in other words, guide RNAs that we think have a high chance of producing substantial off-target editing events. Where all these tools struggle a little bit, though, is actually defining where those off-target sites are going to be. So all of them have issues with their sensitivity and their specificity. I would say that in silico prediction methods are suitable if you don't necessarily need to characterize all the off-target editing events. You really just want to get a sense of whether it's happening or not. But if you need to be able to comfortably say where every single off-target edit is occurring in your experiment, you should probably rely on some of these unbiased methods you see on the left-hand side of the slide. And these are actually methods that have to be done on the bench. So they involve some wet lab work and some sequencing work to generate the list of candidate off-target sites, but they tend to be much more robust. So at IDT, we use a modified version of GuideSeq. This is not a method we developed. You can see the publication for that here. We've made some modifications to it to actually increase the number of off-target sites that we are nominating in this process. Oh, I see a note here asking me to speak a little bit slower, so I'll slow down a little bit for you. So yeah, GuideSeq is a technology or a method we use to actually identify where off-target editing is happening in the genome, right? And so the output of those types of experiments is what you see on the right-hand side here. We basically have a list of genomic coordinates. This top one is actually our on-target site, the site that we intentionally are trying to edit in the genome. And you see that it's a perfect match to the region of the genome we were trying to edit, which makes sense, right? All of these ones below it are off-target sites. And you can see that there's some number of mismatches between those regions of the genome and the actual guide RNA. So the risk of cutting at these sites is probably lower than it would be at the on-target site. But based on the GuideSeq output, we know that editing did occur at these sites. So this is really the output of GuideSeq that we're interested in. And it's what we use to design the rhAMPSeq panel. Now, if you're not familiar with GuideSeq, I've included a schematic here just to show you how this works. Basically what you do is you take your guide RNA with a Cas9 protein and transfect that into a cell line, just like you would if you were trying to edit that cell line. You're going to introduce double-stranded breaks as you would with any CRISPR experiment, but you'll also have co-transfected this double-stranded donor template. So double-stranded DNA, linearized double-stranded DNA, is capable of integrating at these kinds of cut sites, even if there's no homologous sequence on the ends. So that through non-homologous end joining. And that's what this part of the figure down here is showing. Because you know the sequence of this donor tag, you can actually amplify it out by PCR and then sequence that on a next-gen sequencing platform. Once you've done that and mapped these reads back to the reference genome, you have a really robust list of where all of this editing happens. So you'll have reads that originate from your on-target site, as well as some reads that originate from your off-target sites. And that's really how we generate these lists that you see behind the figure. So in order to use rhAMPSeq, we take that list and we input it into our design tool to design a rhAMPSeq library. So let me talk, shift a little bit here and away from CRISPR and talk a bit about how rhAMPSeq works. Because now we're at the point where we've identified our sites, we need to actually understand how rhAMPSeq can help us quantify them. So rhAMPSeq is, like I say, an Amplicon sequencing technology, if you're familiar with that methodology. But what it really boils down to is basically 2 rounds of PCR. The first round of PCR is the one where we're amplifying all of your on and off target sites. And so, you know, the scissors here represent where we would get on and off target cutting in the genomic DNA. And what we will do once you tell us those coordinates is design a pair of what we call RH primers that flank every single one of those sites. And these primers will be designed to be compatible with one another so they can all be used in the same reaction. So that, you know, I just want to make sure that you understand how important that multiplexing is. If you have, you know, 1 on-target site and 100 off-target sites, that means we're going to be putting 101 primer pairs in one tube, and you're going to be running them all simultaneously. The RNA base and blocking groups you see on here are really the reason why we can multiplex that so much, and I'll talk about that in a couple slides. But we can really get robust amplification of many genomic targets this way. So the first round of PCR amplifies the regions of interest and adds the first part of the Illumina sequencing adapters shown in blue and orange. We do a dilution step in between here, so just to reduce the concentration of that Amplicon. And then we do our second round of PCR, where we're adding our sample indices, so P5 and P7 indices, flow cell binding sites onto the ends here. And so this is how we multiplex multiple samples on the same sequencing run. Once you've completed that, you do a cleanup step, quantify your library, and throw it on the sequencer. So all in all, this process is about half a day, and then it goes onto the sequencer. So let's zoom in a little bit more on the primer structure here, and I'll walk through why that RNA base is so important. So here you can see a bit of a zoomed in version of how this works. The large gray bar here is representing your genomic DNA. And here we have the target region that contains one of the on-target sites or one of the off-target sites that we're interested in studying. The first round PCR is done with these rhAMP primers. So we have a forward primer and we have a reverse primer with the SP2 and SP1 sites shown in orange and blue. Now, the real unique part of this application or this methodology here is the blocking group is the green circle and the RNA base in white here. In the master mix that we use to amplify this first round reaction, we include an RNase H2 enzyme. And basically what that enzyme does is detect if there's a match between the RNA base shown here and the DNA base in the template DNA. If the RNA base is complementary to the DNA base, the RNase H2 enzyme will cleave off the blocking group and we get normal amplification, just like any other PCR. If there's a mismatch between the RNA and the DNA, we don't get cleavage, and the blocking group stays in place, and we get no amplification. This is really important for multiplexed reactions, what we're talking about here, because it prevents off-target amplification and it prevents primer diamonds from basically soaking up all the other reagents. And so I'll show this again on the next slide, but that's really the reason why we can have up to 5,000 primer pairs running in one tube. And then the second run reaction is just a conventional PCR that primes off that SP1 and SP2 site to add your sample indices. So just one more figure. I realize this is a sort of a technical concept, so I want to make sure that we're all clear on it. The technology that this is based on, these blocking groups and RNA bases, actually stems back a very long time at IDT. So if you're really interested in getting into the technical details, I would suggest reading this article here from 2011. RNase H-dependent PCR, as we call it here, is actually something that our founder, Joe Walder, was very interested in, has been developing for decades. So we've been integrating this technology into many products over the years. What you can see in the schematic is that cleavage process happening. So we have our blocked cleavable primer with the RNA base. This hourglass figure here is representing the RNase H2 enzyme, which comes in the library prep mix. And so if there's a match or the RNA base is complementary to the DNA base, cleavage of that, and we have an exposed DNA end with a 3' hydroxyl group, which is just like a normal PCR primer. And so then we get extension. This slide really shows you the benefit of that. So I'll focus on the gel that you see on the right here. If memory serves, this was a test panel that we did of about 100 different genomic targets. So the primer mix here contains 100 primer pairs, all running simultaneously. And if we do that with standard DNA primers on an actual genomic DNA sample, you can see that we do get the amplicon that we're interested in generating, but we also get this really intense band of primer dimers. So most of the reagents in our reaction have been sucked up by these dimers, which makes it difficult to efficiently generate the amplicons we want to sequence. If we switch that over to these rhPCR primers instead with those blocked cleavable groups, you can see we almost completely eliminate the primer dimers, and the dominant product becomes our amplicon of interest. We still have to do a little bit of cleanup because you can see there's some large still showing up, but for the most part, a much cleaner reaction. And so that's really the key benefit of rhAMPSeq, that we can eliminate those primer dimers. So in addition to that kind of unique chemistry on the primers, we also take a fairly involved effort to design our primers so that the risk of primer dimers and off-target amplicons is lower. And so I won't spend too much time on this because this is something that actually is quite an automated process. You don't have to worry about it. It happens automatically, but I think it's good to understand it. What you would be providing to us to generate a rhAMPSeq library is basically a list of your targets, right? So we would perhaps have your on-target site and then all of your potential off-target sites. And what our algorithm will do is take those coordinates as well as other parameters of the design, for example, the amplicon length and the desired melting temperature, and it will generate all possible primer pairs that would amplify that region. And it will do this for every single target that you provide to us, so every on and off target site. Once it's done, it will select a single amplicon for each target, and it will test how compatible they are with each other as a pool, right? So we have one amplicon for each region of the genome that we're trying to study. And if it finds that one of those amplicons is incompatible with any other primer pair, it will substitute it out and select a new amplicon instead. And so this is an iterative process that will happen over and over until it's designed a pool of amplicons that is internally compatible with itself. So like I say, that's an automated process, but that's essentially how we build these libraries. So let's shift back to CRISPR a little bit. And I want to talk a bit about how we use rhAMPSeq to do this. This is a sampling of publications from the last few years. Most of them have some involvement from IDT, but some of them are published just by our customers. And all of them are using rhAMPSeq basically to quantify off-target editing. And so we'll share these slides afterwards if you like, and you can read these papers if you like, but I'm going to focus a little bit on this one here circled in red. This was a publication that we co-authored with the Porteous Lab a couple years ago. And what it basically does is characterize how we developed our high fidelity Cas9. And so that's a mutant version of Cas9 protein that is more specific and has fewer off-target editing events. But it's an informative paper for this discussion because the methods described there are essentially what I'm telling you here, right? Using GuideSeq to identify the off-target sites and then using rhAMPSeq to quantify and sequence those locations of the genome. So this is an example of some data from that publication. I won't go through too much of the story here for the sake of time, but basically this was comparing the worst case scenario when it came to off-target editing to the best case scenario of all the conditions that they tested. So the worst case situation was when they used a wild type Cas9 and they let the cell line endogenously express that protein. And you can see that about 3/4 of the edits they detected by rhAMPSeq were off-target edits. And only about 1/4 of the time were we editing at the intended genomic location. If we change this up and move to that mutant high fidelity Cas9, and instead of letting the cells express it, we deliver it as a recombinant protein, so the RNP approach that we recommend, and we almost eliminate all of the off-target editing. I think for the sake of this discussion today, really the message on the right here is the take home. For us to have done this with PCR would have involved hundreds, if not thousands, of individual PCR reactions targeting different genomic sites, and then quantifying the editing at all of them. Instead of doing that, we were able to use one rhAMPSeq library. And so basically generating the data in these figures went from a months-long process to just a few days. So if you're interested in off-target editing, this is certainly faster and generally going to be more economical than all of those individual reactions. And then the final thing that I want to discuss before we wrap up here was just an introduction or sort of a teaser about an extension of this that we're developing and planning on commercializing early next year. And so I've talked a little bit about how you identify off-target sites using technologies like GuideSeq. We've talked through what rhAMPSeq is, and I hope you can appreciate how that can be used to quantify and sequence on and off-target locations in the genome. The problem is what you get out of rhAMPSeq and often Illumina sequencer is just raw reads, right? You need to analyze that data to quantify editing, to get a good sense of what it all means. And so in the paper that I was just talking about on the previous slide, we actually developed an internal pipeline to do this. And we heard from many of our customers that this is a pain point, right? If you're interested in quantifying off-target editing and on-target rates and things like that, analyzing that data is not trivial. And so we've been working to basically move that internal pipeline into the cloud. And so what it will allow you to do is take your FASTQ that come off the sequencer, upload them to our analysis pipeline, and tell us a little bit more about your experiment. You need to tell us the guide RNA and if you were using a donor template. But once you provided that information, you basically hit go and we take care of the rest. So this pipeline is really designed to deal with quite a large sort of laundry list of things that you have to do to clean and process your data. And I won't go through all of these steps, but suffice to say what we're basically doing is taking a whole range of command line tools, which some of you may be familiar with already, that are used to basically clean up and filter your reads, as well as a series of IDT specific tools that we've developed to help bin reads based on the editing types that we're detecting. And we've bundled that all together into a cloud-based analysis platform. The output from this is a whole set of numerical outputs telling you editing rates and things like that, as well as a collection of published and ready images that will be useful if you're trying to disseminate your work and explain where editing is happening and the types of edits that are being detected. So we've done a little bit of work validating this tool, and we're actually in the process of preparing a publication for peer review that should be submitted for peer review in the next couple of weeks. In this particular data set that I'm showing here, what we did was generated some synthetic data, so some artificial data of what editing profiles would be expected to look like for Cas9 and Cas12. And then we asked, of all these analysis algorithms that you see on the bottom, which ones are best able to detect the editing events that we know happened, because this is artificial data, right? So how good are they at actually recapitulating what's happening in the actual experiment? So CRISP alterations, the one on the far right, is IDT's pipeline that we're developing, and the rest of these are ones that you'll find in published literature. A perfect result would be if all of the replicates here lined up on 50. And so you can see different tools have different levels of accuracy, essentially. We think we're doing quite well for Cas9. Some of the other tools like CRISPR also do a very good job. We find that the main deviation is for people working with other CRISPR enzymes, right? So once you move over to say Cas12a, the story is different. CRISPR-SO2 still does really well, but some of these other algorithms are making some assumptions that are specific to Cas9, I think, which is why we don't see such accuracy there. So more to come on this space. And like I said, we have a publication coming out soon on this, but I wanted to share a little bit of data there before we launch that product next year. So just some statements. I won't speak to all of these points, but just to sum it up, we think that rhAMPSeq is a really fantastic way of assessing editing at multiple sites in the genome at the same time. Remember, you can multiplex thousands of locations in a single reaction. And you can also multiplex many samples in one go as well. So protocols that would produce comparable data by PCR are going to take months sometimes, and we can bring that down to just a number of days, depending on your exact experiment. I think everyone should probably think about off-target editing and what it means for your experiment. As I said at the beginning, this is an approach and a technology that for some people really is just not going to be useful, because off-target editing is not the main thing you're worried about. But at some point, especially if you're moving towards the therapeutic space or have that in mind down the road, you're going to be asked about off-target editing. And this is, I think, one of the best tools currently available for assessing that. So with that, I'll conclude. Thank you all for attending. If you have more questions on this, I would certainly encourage you to visit our website. We have a great resource there in terms of recorded webinars and other resources. So this is the URL. For general inquiries, you have our CRISPR email address. I get CC'd on all of those, as well as our application support team. So that ensures that the best person will answer your question. And then if you have any questions about today's presentation that we don't get to in the Q&A, you have my email address there as well.
Adam, I was curious, is there a way to eliminate off-target editing completely, or is this something that just has to be fought?
Yeah, that's a good question. Realistically, I think there's some smart steps that everyone should consider to help reduce off-target editing. The three main ones we recommend at IDT are going to be careful and thoughtful guide RNA design. So lots of tools to help with that, including our own. But really taking a careful look at your guides is the first step. And this is the same as when you would design PCR primers, right? The second is going to be using a method that lowers the amount of guide RNA and Cas9 present in the cells being edited. We know that there's a correlation between the concentration of Cas9 enzyme in a cell and the amount of off-target editing we see. And so that's part of the reason IDT recommends the rNP approach, where you use recombinant proteins and synthetic guides instead of plasmids. And then the third method is considering the use of mutant high fidelity Cas9s, like the one I mentioned in today's talk. Those can substantially reduce off-target editing. So those three things combined go a long way. The reality is off-target editing is probably unavoidable to some extent. So it's always going to occur at least at a very low level.
Okay, great. So if someone is doing an analysis, how many off-target sites should they include?
Yeah, off-target editing is a phenomenon that varies quite a bit from guide RNA to guide RNA. So we have assessed certain guides that produce, we do a guide-seq experiment. We only identify 5 or 10 candidate off-target sites, and the actual experimental data, we might only see off-target editing at 1 or 2 of them. We've also found guide RNAs that in silico we thought would perform well, but produce dozens of off-target events, all of which were detected by rhAMPSeq. And so I think the number of sites that you should interrogate really needs to be dictated by data. And so I would recommend doing a guide-seq experiment up front, just to get a sense of it, and then going from there.
Great. Marshall, are there any hands? I thought I saw one go up.
One went up and it went back down again, so maybe the question was answered, but I have a written question, which I'll read out right now. And the question is, can one check the off-targeting for DCase9 too with rhAMPSeq?
Yeah, so DCas9, just to make sure everyone's familiar, is a mutant version of the Cas9 protein that does not cut DNA, right? Both of its catalytic domains have been knocked out. So it binds to a specific location in the genome, but it doesn't introduce a double-stranded break. So for that reason, GuideSeq and rhAMPSeq are not going to be able to assess DCas9 directly because there's no break being introduced. That said, I think it would be reasonable for you to use a regular Cas9 to get a list of off-target sites and assess editing that way. I think it's reasonable to think that DCas9 is going to bind to the same off-target sites that a regular Cas9 would as well. So my recommendation there would be do the experiment using your guide RNA of interest and assess off-target editing with a wild-type Cas9 and take that list of off-target sites and apply it to your DCas9 experiment.
Okay, thank you. There's a comment and another written question. The comment is, great talk, Dr. Chernick. You explained it so well. Thought you'd like to hear that. And
thank you.
The question comes from Christian Kastrup, who asks, if a researcher wants to do knockout in adult animals, in the liver, for example, does IDT have all the tools to enable this? And if not, what would you recommend?
Yeah, thanks for the question, Christian. The short answer is we have most of the tools. So I think the big challenge with doing genomic editing in adult animals is going to be the delivery, right? So if you're using an RNP, recombinant protein and synthetic guides, the most common approach I've seen people starting to take is to encapsulate those in lipid nanoparticles. That's not something that IDT has any direct experience with, but I do see a number of our customers trying that. And we can certainly make the Cas9 and the guide RNAs at much larger yields, sufficient for animal experiments. So that side of it is something we can support. I do also see some of our customers going this route with viral vectors, for example. We don't produce those sorts of vectors ourselves in-house, but we can provide some support for the reagents you would use to build those, right? So synthetic gene fragments and primers and the like.
Okay, very good, thank you. And I don't have any other written questions or raised hands at the moment. Oh, there we go again. See, I told you as soon as I say that. We do have a written question, which is, what should be the standard or control for rhAMPSeq?
So I think there's a few ways to interpret that question. I think there are a couple controls that are probably smart to include no matter what. In terms of the library itself and the regions that are being sequenced, you should probably include one or two locations that you think are not going to be edited. So these are regions of the genome that you expect to remain wild type. And that'll just give you more confidence that basically the level of sequencing error and sequencing noise coming off the NGS platform is whatever it happens to be. We anecdotally typically say that's about 1% of reads have some spontaneous mutation or substitution present in them, but including a couple sites that you expect no mutations in will help you actually quantify that. I think in terms of experimental design, it is probably good to run the rhAMPSeq library on an unedited sample as well, so that you can get in a sense of how well are all of those amplicons actually being generated. When you run the library protocol, we take a lot of steps, and I hope you can see that in the presentation, to design good panels with primers that we think are going to work very well, but those are all in silico predictions, and occasionally one particular loci won't amplify very efficiently. That's something that you would see if you use a wild type control, unedited control, and can account for in your subsequent analysis. So I hope that answers the question, but feel free to submit a clarification if you had something else in mind.
Okay, thank you. And presently no further questions.
Great, so I can jump in again. So I was curious, how long does the whole process take from identifying the off-target sites to having the data in the final analysis?
Yeah, and that's a sort of a multi-step process. Nomination of off-target sites using something like GuideSeq, that's a protocol that's probably going to take a week or two. Fortunately, it's something you only have to do once with a particular guide RNA. So once you've done GuideSeq with a guide RNA, you have your list of off-target sites. You don't necessarily need to do GuideSeq again unless you decide to use a new guide RNA. So once you have your off-target site, you can submit the design request to IDT. Typically, that takes us less than a day to design it, and if you order it, we typically will synthesize these in under a week. And from there, it's a half day of bench work for you to prepare the library, and then usually half a day of sequencing time, depending on the sequencer you're using. So I think once you have all of your reagents in hand, we're only looking at a couple of days to get your data and then analyze it.
Great. And then does rhAMPSeq or an NGS have to be used for assessing these off-target methods, or are there other methods that can be used?
Yeah, and there are actually quite a range of methods we see people use for assessing, especially for assessing editing at the on-target site, right? Because that's a known location in the genome. And so whether you use something like a T7E1 assay, which is just a quick check of whether editing happened or not, or something like a qPCR assay, which can quantify the amount of editing but won't necessarily give you sequencing data, there are a few options for that. And of course, you can extend any of those methods out to more than one genomic location. So you could use something like qPCR to check for off-target editing. I think the tipping point when NGS starts to make more sense is when you begin to look at multiple sites simultaneously, right? And so in some of the examples I gave, the slide from that publication had about 30 off-target sites shown in it. Running 30 separate qPCR reactions in parallel is expensive because you need all those reagents and time consuming. And so at that point, NGS starts to make more sense. So it really depends on your expertise in the lab and your access to something like an NGS system. But I think for most people who begin to look into off-target effects and really want quantification, NGS is probably the best.
Okay. And then I'm curious, when you're doing this in cell culture, are there any effects that you can see when you get off-target editing?
So, the consequence of off-target editing in cell culture will vary. Sometimes we really don't see any real phenotypic consequence. You have to remember, as cells divide, there's always small mutations coming up. Some of them have an impact and some of them don't. I think occasionally you will see an off-target effect, particularly if it happens to happen in another gene that can result in fairly severe phenotypes. So, failure of cell culture to grow, or if you're doing this in a model organism, maybe a lethal mutation embryonically, things like that. So it's really going to vary. I think the biggest application of this type of methodology is going to be, in coming years as we see CRISPR applications moving into the clinic. Regulatory bodies are going to want very robust ways of measuring this type of, effect at the genomic level. And so that's really the intention of building these tools, is to give scientists a good way of just knowing what's happening in a genome-wide sense.
Great, thank you. Marshall, are there any hands up or questions?
Yeah, I've got another written question I'll read out for you, Adam. And the question states, I'm curious why or how high fidelity Cas9 is more accurate than Cas9.
Yeah, that's a great question. So if you know the history of high fidelity Cas9 mutants, you may be already aware of the fact that many of them have a kind of an inherent flaw, and that is, while they reduce off-target editing, they also reduce on-target editing. So the only reason they're more specific is probably because they're less active overall. And so, that's not the end of the world if you can get enough editing to do whatever you want to do. But we've seen quite a few cases where you take some of these high fidelity mutants, transfect them into cells as rNPs, and really see no on-target editing. I think that's really the objective behind why we developed our hi-fi Cas9, that we try to maintain a good level of on-target editing. If you look in that publication I showed in my presentation, we actually do say exactly what the mutation was. It's a single amino acid substitution. We don't exactly understand call it the biomechanics, if you will, of why it's more specific. We haven't done things like crystallography of the different states the enzyme goes through when it's cutting. But we have generated a substantial amount of data showing that it reduces off-target editing while maintaining a very similar level of on-target editing. And for us, that functional consequences is the most important thing. I think in time, we'll see more and more work on the biochemical side of this to better understand why off-target editing happens. And that should also help clarify why this particular mutant reduces that effect.
Great, thank you. There's no other written or questions or raised hands at the moment.
Well, in that case, I will ask one more question and request a final round of questions for anyone who'd like to get any in. My final question is, if someone is interested in working with IDT, Adam, are you the right person to reach out to initially, and you'll get them sent in the right direction, or should they reach out to someone else first?
Yeah, I'm happy to be the person you reach out to, especially if it's at all CRISPR related. In worst case, I'll find someone else to help you if it's not my area of expertise. Many of you will also already know Bob Setter. He's your local sales rep, and he lives just outside of Vancouver. So he's a great resource for anyone on the West Coast. And then we have local sales contacts on the eastern side of the country as well for our attendees out there. So yeah, feel free to email me. I'm actually based in Calgary and have a lot of experience with our Canadian sales team, so I can put you in touch with the best person based on your geography.
Great, thank you. Marshall, any final questions?
Indeed, there is. What is the recommended mixing ratio by IDT for Cas9 and sgRNA?
Yeah, so most of our protocols recommend basically a 1-to-1 ratio of protein to guide. You'll see a little bit of variation in that, just based on how volumes that are convenient to dilute things in, basically, but essentially 1-to-1. We have experimented with other ratios of that, and I think if you look through the literature, you'll see a broad range of anything from 4-to-1 to 1-to-4 is kind of what I've seen. We haven't seen a ton of benefits to those other ratios, and intuitively, my thinking is that that's a bit of a waste of reagent, right? If you use excess, Cas9, for example, well, if there's no guide RNA to bind to that Cas9, it really doesn't have an opportunity to do much of anything. So yeah, for that reason, most of our protocols recommend 1-to-1.
Perfect. So thank you so much for joining us today. So if you're interested in learning more about rhAMP, CRISPR, or IDT, please feel free to reach out to Dr. Chernick